For many people, the concept of evolution can be captured in the image of a fish struggling up onto a muddy bank. It signifies nature's awe-inspiring conquest of land and reminds us that we all started out, quite literally, as fish out of water. However, the fish that originally left the water roughly 370 million years ago were fundamentally different to any fish alive today. They did not have fins, but limbs, and that transition from fin to limb is one of the most dramatic and important events in evolutionary history. It should first be noted that terrestrial locomotion was not the original purpose of limbs. Studies of transitional fossils have shown that limbs were likely first used to prop the organism's head up out of the water to aid with air breathing. Secondly, I should define exactly what is meant by fish in this context. Almost all living fish belong to the clade of actinopterygians, or ray-finned fish. However, tetrapods are more closely related to lungfish. Along with coelacanths, tetrapods and lungfish make up the sarcopterygian clade, otherwise known as the lobe-finned fish. Another subset of the sarcopterygians is the tetrapodomorpha. This group is defined by all the fossil lobe-finned fish that are more closely related to tetrapods than they are to other living lobe-finned fish. It is one of these transitional tetrapodomorphs that took the first steps onto land roughly 370 million years ago. Looking at the overall body plans of tetrapodomorphs, the transition from fin to limb becomes more clear, and examining the bones of the limb in detail shows a gradual movement towards the stylopod, zygopod, autopod pattern seen in tetrapods today, with a humerus, radius, ulna and wrist visible in Tiktaalik. Acanthostega's wrist is thought to have been cartilaginous, and, unlike Tiktaalik, it has digits. Analysis of Acanthostega has shown that its limbs were likely not weight-bearing, meaning that the evolution of digits preceded the migration to land, and not the other way around as previously thought. Furthermore, it does not take an expert eye to determine that Acanthostega has more than 5 digits. This challenged the theory that pentadactyly is a primitive trait. The explanation for complex segmentation, such as that in Acanthostega's digits, can be found in the developmental genetics of modern tetrapods and their ancestors. Developmental genetics is a powerful tool for researchers studying the evolutionary history of limbs. Both fins and limbs begin as paired swellings or buds of the mesenchymal cells in the ventral body wall, with an external layer made from ectoderm. In tetrapods, this is known as the apical ectodermal ridge. Limbed and finned animals also share a structure found to the posterior of the bud known as the ZPA, or zone of polarizing activity. The ZPA expresses the sonic hedgehog gene, which is involved in determining the identity of digits and fin rays. This is a very highly conserved process found in all living jawed vertebrates. In tetrapods, as the limb bud grows, the more proximal cells begin to form the more proximal segments of the skeleton. This segmentation is driven by Hox genes. For example, if you knock out HOXD13 and HOXA13 from a mouse embryo, its limbs will develop with no wrists or digits. In fact, it is thought that incomplete expression of HOXD13 in Devonian tetrapods such as Acanthostega is what drove their development of so many digits. We can see from the fossil record the morphological changes that defined the fin to limb transition, but it is equally important to investigate the genetics that drove those changes. One question posed by researchers was whether tetrapod Hox genes have homologs in ray-finned fish that determine the orientation and segmentation of the fin skeleton as they do in tetrapod limbs. In tetrapods, Hox expression comes in 2 phases during limb development. The early phase is involved in the formation of the stylopod and zygopod. The late phase develops the autopod. For a long time, researchers could not find enhancers that linked Hox genes to late-stage development of the fin in actinopterygians. However, genomic analysis of paddlefish shows 2 phases of Hox expression, as in tetrapods. This is because the paddlefish is a very basal ray-finned fish. The earlier experiments used the more crownward zebrafish, which is a teleost. One of the defining features of teleosts is that they underwent a whole genome duplication event. It is thought that this dramatically reduced the expression of the late-phase Hox enhancers, making them much more difficult to find. However, after the broad homology was discovered in paddlefish, specific early and late enhancers were found in more crownward ray-finned fish. Their expression pattern is very similar to their behaviour in tetrapods. The early phase enhancers are expressed across the whole fin bud in early development and then disappear. The late phase enhancers are found only in the distal parts of the fin. This supports the theory that the distal fin bones are homologous to the autopod bones found in modern tetrapods. In conclusion, a combination of paleontology and developmental genetics has provided a deep insight into the fin-to-limb transition, improving our understanding of our own species and the others with whom we share the planet.
Molecules and particles from follicles is fossil fuel. Maybe it's impossible to change things. Zika or Ebola.